For our project, we looked at the inner structure of oranges and broccoli and cauliflower, looking for self-similarity within the elements.
We framed our study of broccoli and cauliflower within the discourse of fractal geometry in biology and architecture. Mandelbrot, the pioneer of this field, explained that the self-similarity in fractals arises from a series of transformations at different scales, where each level has a specific level of diversity. For this reason, the more appropriate term to describe these structures would be self-affinity, where variations at each cycle make the final overall composition. Stan Allen, in his book From Object to Field, explained that the relationship from part to whole is essential, but what is really in play are the tensions between local difference and overall stability. The broccoli and cauliflower are classified in the Brassica oleracea species. In scholarship, it is commonly known that Romanesco broccoli or Romanesco cauliflower is a fractal structure. For this reason, we wanted to compare other kinds of broccoli and cauliflower to see if they expose similar internal structures. We examined Calabrese broccoli, white cauliflower, and purple cauliflower. We distinguished between the flower heads and the stem, mainly focusing on the flower heads to see if the images at higher magnifications showed a similar structure as the real-life image we see. The following image shows Calabrese broccoli. These were used to frame the research on Brassica oleracea. By comparing this structure to the next image of white cauliflower, we can see that the buddings at the extremes of the stems are similar. These images show a clear similarity between the flower heads and the inner structure. However, the visual resemblance of these images is not sufficient to determine whether this type of cauliflower is a fractal. We then used the confocal microscope to take our study one step further. We built a digital 3D structure of images taken from purple cauliflower, testing whether or not we could consider them fractals. Although they are similar, we were still unable to conclude they were fractals because we would have to study the structure at larger and small scales. With only two scales to compare, we can only say that the structures are visually similar.
The orange is classified as a hesperidium, or a berry with a thick rind. It has four components: the rind, the thick outer shell, the pericarp or albedo, which is the white pulpy inside of the outer rind, the pith, which is the white material attached to both the inner rind and the orange slices, and the juice sacs that comprise each slice. With the orange, we were looking for some small-scale structure that would indicate a communication or interaction between the four components. We found one similarity across all four. It seemed that each had a pore structure, which might indicate the passage of water or other materials between the four components. The outer rind looks porous. This is 20% magnification, or 20 times magnification. The pericarp did as well, while the pith appears to have liquid pockets integrated into it. The individual juice sacs have spots of them, which may be some type of pore in the membrane.
Thank you, and I hope you enjoyed our presentation.